Hello, everyone, and welcome to the WTR Healthcare Happenings podcast. I'm your host, Shawn Severson, CEO and founding partner of WTR Research. In today's podcast episode, I'm being joined by Dr. Paul Sargeant, President and CEO of Circular Genomics, and also joining us is WTR Research's healthcare analyst, Robert Sassoon. Circular Genomics is a private held molecular diagnostic company focused on developing next generation blood tests using circular RNA biomarkers to enable earlier detection, clearer disease characterization, and more precise care for neuropsychiatric and neurodegenerative conditions. By integrating proprietary biology, advanced analytics, and clinically focused development, the company aims to transform how neurological diseases are diagnosed and managed. So first, let me welcome both Paul and Robert to the podcast.
Very nice to meet you both.
Great to meet you too, and thanks, Shawn. Great to be here.
Thanks. And Paul, before I hand it over to Robert for his questions, I just wanted to start with one and ask you to tell our listeners about your professional background, what brought you to Circular Genomics, and then the 30,000-foot overview of the company.
Absolutely. Again, really nice to meet you. Thanks for the opportunity here. I was born in London and always had this interest in the application of science and technology to help humanity. I think you can look back at the history of science and tech and see how it really has helped humanity to thrive. I did a PhD in the biological sciences, University of Cambridge. I haven't got the patience to be a full-time researcher. And so I spent most of my career at the interface of science and technology and its funding, IP expansion, productization, product launches, all the way through to exits. A number of roles with companies like Bio-Rad, Medtronic, and Thermo, 7 buy-side and 3 sell-side transactions. Sort of a high-level overview of circular genomics, what attracted me to the company and to this opportunity. I think we've all seen the incredible benefits that precision medicine has brought to oncology and cardiovascular. And I think somewhat tragically, we can't say the same thing yet for neurology, neuroscience, and mental health and brain health issues. So really, it was a desire to bring a deeper molecular understanding, because that's really where you begin to unpack what is happening with the organ, with the brain. Perhaps our most important organ, you could argue. It makes us who we are, what we are. We've all been, no doubt, affected in our families, our loved ones, by mental health issues, and sadly by brain health issues, Alzheimer's, Parkinson's, etc. And so we do have an opportunity with the circular RNA biomarkers, this really esoteric endogenous RNA species, to understand much more fully what is happening inside the brain, but having that understanding not by probing the brain directly, but through what is needed for scalable diagnostics and prognostics, being able to access those biomarkers in the blood. Again, a quick summary of the company. Founded in 2021, we've secured a foundational IP estate related to the use of circular RNA biomarkers in Alzheimer's and elsewhere. Developed key data sets, and I think unusually for a company at this stage, in a very high number, over 2,500 patient samples at this time, with solid replication, built a world-class SAB and commercial advisory board. We are located in Lilly Gateway Labs, which is part of Lilly, the drug company, in San Diego.
Great. Thanks for that overview, Paul. Very helpful. And Robert, I'm going to turn it over to you at this point.
Thanks, Shawn. And Paul, good to have you on this podcast. So let's delve first into a little into the Circular's platform and technology. So could you start with a brief overview of circular RNA, including what biological properties make it particularly attractive for diagnostic use, and what advantages it has to offer over traditional linear mRNA or DNA-based liquid biopsy approaches, particularly in terms of diagnostic sensitivity and stability?
Yeah, great question. But really a key question, right, in the diagnostics and biomarker segment. So circular RNAs were discovered back in 1979. Sometimes the path to a biomarker development takes a long time, as you can see from that extensive timeline. Circular RNAs are endogenous, naturally occurring, closed loop, non-coding RNA. Around 100,000 circular RNAs have been characterized predominantly in the brain. So the expression profile is predominantly in the brain. It will be useful and instructive to view circular RNA biomarkers as being the conductor of the genomic orchestra in the brain, this dynamic brain biomarker. They're involved in activities such as gene regulation. They act as a microRNA sponge, binding, sequestering proteins, and regulating transcription or splicing. Their overall attributes make them an ideal neurological biomarker. So this overexpression in the brain, their very close association with several diseases we know of, their association with all known brain processing physiologically and pathophysiologically, the closed loop nature, given the name circular RNA, the 3 and 5 prime end covalently loop, forming an approximate circular structure, making them very stable and robust. So unlike microRNA with a short half-life in the blood, circular RNAs have a very long, extensive days to weeks half-life in the blood. So the physiological enrichment in the brain, the long half-life, the fact that they cross passively the blood-brain barrier through the CSF into the blood and can be picked up with a relatively simple blood test, again, make them this ideal neurological biomarker.
Okay, but so given the RNA's low abundance relative to linear transcripts in blood samples, how has circular genomics overcome this to achieve sensitive, scalable detection?
Yeah, absolutely. So unlike other species of RNA, like microRNA, subject to exonuclease degradation, circular RNA avoids that. We've perfected a sample handling, RNA extraction, circular RNA identification using AI machine learning methodology, and that's how we're able to overcome this intrinsic limitation. Circular RNAs are there in the blood for a long time, and so that's why you can pick them up with this enhanced sensitivity and specificity.
Got it. So let's turn to your pipeline. Could you walk us through your current pipeline and the development stage of each program that you have?
Absolutely, great question. So, like any company, I think you begin with what is most tangible and real and immediate problem. So, we developed an assay called the MindLight assay. In that particular situation, it was a specific circular RNA we identified highly associated with the response to an antidepressant, SSRI. We developed that all the way through to LDT. But then, as you know, other opportunities arise, such as Alzheimer's, Parkinson's. So we reassessed the market. We decided to make the MindLight assay available for out-license or even divestiture. We're in late-stage discussion with an external party related to the MindLight assay, which is already at LDT, already with a good evidential basis, already with a key publication in place. We're in discussion with that party for, as I said, the out-license potential for the MindLight asset. The Alzheimer's research use only assay is in development. That is slated for a launch later in 2026. That really allows biopharma in particular, but also academic partners to enrich their cohorts in clinical drug trials using the benefits of this circular RNA biomarker-based understanding of biology and pathology. So we're launching that RUO-based assay, which is not an uncommon approach in this space, going RUO to LDT to ultimately FDA submission. We're de-risking the future via that initial RUO path, already in quite late stage discussion with several parties to gain access to this research use only service. Think about that RUO offering in terms of a clinical trial cohort enrichment, identifying the absolutely correct AD patients early enough and precisely enough.
Yeah, you mentioned MindLight, and you've switched. It seems our strategy has switched a little, focusing more on the neurodegenerative disease. Why is that, and how did this test, MindLight, actually address a clinical unmet need in depression patients?
Yeah, great question. So if you think about the first place to begin when you're developing any biomarker test, that is the clinical workflow, the standard of care that exists today. So tragically, and I think we saw this a lot through the COVID pandemic, where unfortunately rates of depression increased for obvious reasons quite dramatically. Patients who are diagnosed with depression, generally speaking, the first course of action is to be prescribed an antidepressant. Those antidepressants, typically an SSRI in the first instance, first line therapy, fail in 60% of patients. That's a very high failure rate. We wanted to develop a test which ensured that more patients get onto the right therapy sooner. So could there be an early reflex to an SNRI or an atypical antidepressant? Because you know that the SSRI is not going to work. So that was the clinical evidential standard of care basis for developing the MindLight test. And again, as you know, in a startup venture, you're always looking for the optimal, highest value creation opportunity. With the MindLight assay, we thought that was best positioned to an external party to commercialize and take further. And we focused on the highly differentiated Alzheimer's and Parkinson's space.
Got it. Understood. So turning to your Alzheimer's program, while amyloid beta plaque accumulation has been the initial focus of disease-modifying therapies, emerging research highlights multiple contributory causes and progression drivers. So how does your RNA-based technology fit, with regard to the Alzheimer's program, fit into that playbook?
Absolutely. Again, that is a fantastic and prescient question, and the answer is going to be somewhat long. So bear with me. So, like any progression, I would say in medicine and science based upon biology, you begin with an initial thesis. So, what do we understand about this disease condition, the physiology, the pathophysiology? And for Alzheimer's disease, that has been the amyloid tau model. So amyloid plaques and tau tangles, those two being related. There's been the exploitation of that model has resulted in amyloid clearing drugs, and certainly the development of p-tau and p-tau 217 based diagnostics in the space. But I think even within the community, the Alzheimer's community and industry bodies and funders like the ADDF, there is high cognizance of the fact that understanding, that biomarker-based approach, amyloid tau, is necessary but likely insufficient. So there are other factors, so amyloid accumulation, tau tangle formation, but also neuroinflammation and neurodegeneration. So where circular RNA biomarkers intersect and impact that current model is that we also identify amyloid tau pathology, but we also layer in that systems level view of biology. In addition, we layer in synaptic dysfunction, neuroinflammation, metabolic issues, oxidative stress, and so forth. So what we're doing is taking the amyloid tau model and expanding and refining upon it, not abandoning it, but expanding to give a systems level view of biology. We know, for example, that if you focus on amyloid pathology alone, in around 30% of patients who you see amyloid positivity, they do not, in fact, ultimately develop Alzheimer's disease. So again, that speaks to, I think, the incompleteness of the model. It's not a bad place to begin science advances in this manner. But with these unique circular RNA biomarkers, this ideal neurological biomarker, we developed this systems level view of what Alzheimer's truly is at a biological and pathological level.
That's really interesting. So let's move a little more to the corporate level. How important are collaborations with pharma or large diagnostics companies in advancing both your clinical programs and the broader circular RNA ecosystem?
Yeah, absolutely. So for a venture-backed startup, you obviously want to leverage the expertise and the network of larger biopharma companies and diagnostics company. As you can expect, we're in discussion with all of the above, gaining access to their expertise, gaining access to sample sets they have available, gaining access to their commercialization channels and platforms. On the biopharma side, as I mentioned, improving patient selection, accelerating enrollment, enabling more precise measurement of treatment response. That's our path with biopharma and in discussion with multiple parties there already. On the diagnostic side, partnerships allow us to scale quickly, increases capital efficiency by leveraging established infrastructure. It really is a goal. I think part of the mission and the very personal vision here, having lost my own father to Alzheimer's about 18 months ago, that we want our tests to be in the hands of as many researchers, as many labs as possible. And so having these diagnostic partnerships and commercial channels allows us to scale more quickly.
Sorry to hear about your father. Now, you've raised $19 million in total across 5 rounds and recently closed their $15 million Series A and are working on a Series B, I believe. As the company advances towards commercialization, how are you thinking about capital allocation, balancing clinical validation, platform expansion, and commercial infrastructure?
Yeah, again, that's a great question. And it's never an easy balance or harmony to attain. At this stage for the company, we would view that data, clinical information, analytical and clinical validation are the currency for the company. That is the predicate upon which we will raise further money and enter into partnering relationships. So there is a disproportionate emphasis, as you would expect, in capital allocation towards that area today. I would say 45-50%. With the launch of the research use only service that I mentioned to biopharma and academia to generate our first revenues in early 2027, there will be a shift to more of a commercial focus on the spend. And we will ramp that up into 2027 as we want to gain access to increased marketing, positioning, promotion of the RUO service, and also in preparation for the LDT dossier preparation and launch of the LDT in early 2027.
So talk about commercialization. What is your nearest term commercial path, and why did you choose that path? And what further phases of commercialization are there, and how will you balance focus, financing, and execution across these?
Yeah, great question. I mean, I think there's a broader aspect to the question. It's again very, very prescient question, particularly in the diagnostic space, with VC inflows to the diagnostic sector being a little depressed compared to prior years. I think you have to be really cognizant of what is that de-risked path to full ultimate reimbursement via an FDA test. So that is the ultimate end game for a diagnostics company like us, a biomarker-focused company like us with this incredible data set in Alzheimer's and emerging into Parkinson's disease also. The initial launch, as I mentioned, is a very de-risked path, working with biopharma companies and academia in providing this a very valuable circular RNA-based signature of Alzheimer's disease, both confirming Alzheimer's disease in symptomatic patients and also identifying in the earliest possible manner the true progressors to Alzheimer's disease up to 5 years prior in pre-symptomatic patients. We will begin with that RUO offering this year. That de-risks and begins to generate revenue the LDT launch. The lower regulatory burden on LDT allows us to develop that, as I said, into early 2027 and begin to generate further revenues there. So two earlier revenue generation paths before we get into FDA submissions. We did develop a very prestigious world-class commercial advisory board because the commercial questions are, of course, just as critical as the scientific questions. So we have the SAB and the CAB working in concert, but we're very cognizant of the challenge of the pre-submissions, working with payers early so that we develop the right strategy, the right data set, clinical utility, clinical evidence generation, so we attain that ultimate reimbursement.
Thanks for that. Final question from me. Does your roadmap include a potential exit strategy at some point in the future?
Absolutely. Whenever I join a company, again, being a growth phase to exit centric CEO as opposed to a founder CEO, lacking that patience. I think when you join every venture, you want to have a view to what is the ultimate optimal endpoint for the company as you drive value creation, which is a predicate for that ultimate exit potential. I think the likeliest exit potential for a company like us with this novel biomarker platform, this de novo set of data related to Alzheimer's, expanding into Parkinson's, that they share a common set of pathways, would be a bolt-on technology acquisition by an established diagnostics party. That really is also in line with our vision for the company, that we really deliver precision medicine in neurology. I, as a venture-backed startup, have probably an intrinsic limit to the build of the size of the sales force that I could generate. Working with an appropriate diagnostics partner, with channel, with infrastructure, with brand, I can make sure we get this incredible test into the hands of the right parties to make a difference to clinical outcomes. So that is the intended exit, which we're focused on even now, every day.
Well, thank you, Paul, for guiding us through Circular Genomics and its business model. And I wish you all the success, and we'll definitely be monitoring your progress. So I'll pass it back to Shawn.
Great.
Thank you
Thanks,
so much,
Robert.
Bob.
And thank you, Dr. Paul, for joining us today. Thanks to my equity research colleague, as I mentioned, Robert Sassoon, as well. As a reminder, please subscribe and follow our podcast. You can also access all of our research at www.watertowerresearch.com. That's open access for all investors, and I'd encourage you to take a look. Finally, a special thanks to our editor and producer of the podcast, Krista Fitzpatrick, and thank you everyone for joining us today.
Thank you.